molecules that get the rest of the adaptive immune system going too. So if this cell doesn't get stimulated because you don't have enough of them because you have HIV or because you're undergoing chemotherapy, then you can actually not be able to do a good job stimulating the adaptive immune system. So when we talk about people who are immunosuppressed, or we talk about the very old or the very young, people whose immune systems aren't working well, oftentimes we're referring to this process, sometimes very specifically to this process in the case of HIV. Amos.
So also in case of AIDS, so it's
HIV, AIDS, yes.
Yes,
Yes, AIDS is what HIV causes.
Okay.
Exactly. All right, how are we doing on this? Does this make sense? I'm looking in the back for people nodding. Yes? Okay, we're good. All right. So helper T cells, once they get activated, magic happens in your immune system. All right, so here again is the same picture, except for I was too cheap to buy the picture. So see, it says D. Anyway, it's all good. Really should just buy the picture. Anyway, so here's antigen recognition. So here we go again. Here's our antigen presenting cell. Is it MHC class 2 or class 1 or both?
Both.
It's both, right? So here we're phagocytizing the antigen. We're chopping it into little pieces. We're presenting it using MHC class 2. We're showing it to a helper T cell. The helper T cell goes boing, I'm getting activated. And then, right? And then what it's going to do is it's going to do all these really exciting things. So it's going to divide and make copies of itself. These are memory cells. This means that the next time you see this antigen, your immune system's going to say, oh, that we've seen you before. We are so ready for you. Divide. And the cells are ready to go because they're going to circulate through your body, hopefully for the rest of your life. All right, now depending on how strong your immune response, this is more or less true, but these should circulate, hopefully give you lifelong protection against this antigen. Which is useless in the case of a common cold, because the cold's just going to change shape and you're going to have to start all over again. All right, in addition, this helper T cell is going to make copies of itself and to circulate long term, but it's also going to stimulate effector cells. So it's going to stimulate and activate killer T cells, which kill infected cells. It's going to stimulate B cells to make antibodies, and it's also going to help stimulate other parts of the immune system. So remember we said that the adaptive immune system and the innate immune system, we talked about them separately, but they're really connected. And one of the ways they're connected is because antigen-presenting cells are part of your innate immune system, but they communicate to the adaptive immune system. And it goes the other way too. Once T cells are activated, they make molecules that stimulate things like neutrophils and phagocytic cells to get them more active. So it becomes kind of a circle. They're talking to each other. They all are related, okay? So this T cell in the adaptive immune system is going to stimulate your innate immune system as well. Okay, just to kind of keep some of these ideas going here. A cell that is part of your liver would contain class 1 MHC, class 2 MHC, both and neither. All right, last chance to click in. Okay, so it's a liver cell. What's its profession?
Filter.
Being a liver cell. Yes, filter. Don't get too specific on it to show off your anatomy. I just wanted you to say liver cell, but you're right, it does filter things. Okay, so the answer is that it's not a professional antigen presenter, right? So only professional antigen presenters have class 2 MHC. So it contains class 1.
Yes.
For those of you, which was a lot of you that missed this, does this make sense now? So, I'm getting no feedback. Okay. So class 1 MHC are in all your nucleated cells. So liver cells will have class 1. But only the professional antigen presenters have class 2. Only dendritic cells, macrophages, and B cells. Any other cell in your body will only have MHC1. Okay? Because what that MHC class 2 is for, it's the steak, to put the head on the steak. All right? And if your job isn't to put heads of bacteria on steaks, which your liver cell that's not its job, then you won't have class of 2 MHC. You won't be expressing it. All right, we're trying again. A dendritic cell would contain last chance to click in. Boy, I didn't agree to stopping in here. All right, so this time you did a little bit better. It's going to contain both, right? Because it's both a nucleated cell and a professional antigen-presenting cell. So if you're struggling with this, which a few of you are, this is definitely something you want to review. And feel free to email me if you want me to try to find you some links, or maybe I can find a Khan Academy video or something that will help kind of go through that. I hit this hard because I think it's so important. It says name 2 antigen-presenting cells. Okay, we know 3. What are they?
Macrophages, dendritic cells, B cells.
B cells, macrophages, dendritic cells. Okay? What is their role in the adaptive immune system? Well, what's their profession?
Vaccination.
They present antigen. Okay? Once we talk a little bit more about B cells, you'll also be able to say they both present antigen to T cells, and then also they make antibodies. Which classes of MHC proteins do antigen-presenting cells express? 1 and 2. They express both of them. Okay? And it's important because they're nucleated cells. All nucleated cells express MHC1. It's how they say, it's just me to your immune system. But the professional antigen-presenting cells need that because that's the stake that they're going to post things on. The second question says, how do antigen-presenting cells activate T cells? And here's what I want you to do if this question shows up in the exam. I want you to draw that picture that I showed you here. And I think that being able to draw this on a blank sheet of paper and explain it to yourself is a really good way to study for a big chunk of the exam. Okay? You need to be able to tell me that an antigen-presenting cell is going to phagocytize the pathogen. It's going to chop it up into pieces. It's going to post its head on a stake, the stake being MHC class 2. It's going to show it to the right T helper cell with the right shape that binds to it, causing this cell to get activated. The activation of this cell results in it making clones, copies of itself, some of which are going to become membrane cells to circulate and give you long-term protection. And then it's also going to make chemicals that activate other cells in the immune system to help you fight off the disease. But drawing a picture can be a really good way to do that. And one of the really nice things about drawing a picture is that you can look at this and then you can cover it up, and on a blank sheet of paper, it can be a paper towel, like if you scratch paper, it can be a phone bill back that you're about to recycle, right? You don't need to use fancy paper. Try to draw it and tell yourself the story. When you get stuck, go back and figure out where you got stuck. Cover it up again, start again. 2 or 3 times of that, and I think you're going to find you know it really well because you're telling yourself that story. Teach it to your mom. If that's too hard, teach it to someone who has to listen to you, like, your significant other. Withhold favors, like cooking for them. I'll cook your dinner, but first. All right, but find a way to, teaching is a great way to learn things. If you really want to know what you don't know, try explaining it to someone, and you'll figure out really quickly which parts you didn't understand well. It happens to me all the time. So don't feel bad, but keep practicing until you've got it down. Looking at it and saying, I think I know it, and taking the test is not a good plan. Because it's way harder to explain it than it is to look at it and think you know it. And on this test, you want to be able to explain it. Because that's what a test is. It's asking you to show that you can explain it to someone else, that you've learned it at that level. Okay. So B cells and T cells. So B cells are your cells that produce antibodies. So we've already talked about antibodies. So when antigens are in the body and T helper cells get stimulated, they will stimulate the B cells that have just the right shape to attack those antigens. And we talked about what antigens are. And we said that antigens are really just molecules that stimulate antibody production. And another way to refer to them is that they're immunogenic. So there's a few words here. Something that's an antigen that's very immunogenic is going to stimulate the production of a lot of antibodies. All right, and this is a review. Yes, Charles?
So B cells can stimulate T helper cells and
Yeah.
vice versa,
Well, they need each other.
depending on who gets the antigen first.
They need each other. So what happens is the B cell will actually present the antigen to a T helper cell. The T helper cell will decide whether or not it's an appropriate antibody to be made. So it's part of that, making sure that you don't make antibodies against yourself job. It does that role. And then it will make chemicals that will then stimulate the B cell. So they actually have to talk to each other. So the B cell presents antigen, and that's how it gets the chemicals from the T helper cell to be stimulated. All right, so here's our clonal selection again. Here are our B cells. And again, where we have a pathogen, here's our little pathogen. Remember that you have millions of these in your lymph nodes, but this pathogen only binds to this particular naive B cell. And that's going to cause it to make some memory cells. It's going to begin to clone itself. It's going to make some memory cells to circulate long term. And some of these will then differentiate into a plasma cell. And now instead of the antibody being attached to the surface, it's going to secrete antibodies into the blood. And that's awesome because now these antibodies in the blood can do all of those things that we showed on that slide. They can neutralize pathogens. They can agglutinate pathogens. They can decorate them and opsonize them, like dipping them in guacamole and make them more vulnerable to phagocytosis. Okay? So these plasma cells then are mature B cells that are secreting antibodies. The antibody is no longer attached. It's isotypes like IgM or IgG or IgA that are going to get secreted out into the body. Primarily in the bloodstream, we have IgG. So these become, we call them circulating antibodies. They're not attached to cells anymore. They're free to go and attack pathogens all over your body. Okay. So Charles, this gets to your question. Normally, B cells require a T helper cell to be activated. So how does this work? So here's our extracellular antigen. Remember that we said that B cells were MHC class II. So that means that when a B cell binds to an extracellular antigen, that it then goes and it presents it on the surface and it finds a T helper cell. And it says, hey, what do you think of this? And the T helper cell says, hey, nice work. And it makes cytokines which are going to help this turn into a plasma cell and secrete antibodies. All right, so there is a binding that has to happen between the B cell that is bound to antigen and the T helper cell. Okay? Once this plasma cell is activated, then it will secrete lots of antibodies that will bind to these antigens on the surface. Okay? All right, so here we go in cartoon form, because everybody learns differently, right? Here's our B cell. He says, he's got a B on his hat, so you know he's a B cell. He says, an invader. I must inform a T helper cell. Stanley died this week, right? It's appropriate. For those of you that are Marvel comics. Hey, T helper cell, look what I found. Here's our T helper cell. He's like, see how he's presenting it to him? See that? Antigen presentation. He says, good job, B. It looks like non-self. And he's like, yes! And then the T cell says, here you go. Here's some cytokines to get you activated. And now look. He's like, oh, I'm activated now. So now he's going to make memory cells and he's going to make antibodies, differentiate the plasma cells and make antibodies. Whatever works. I can show you a porn video. All right. Just to make things more complicated, slightly, I do want to mention that there are a few antigens that can activate B cells without T cells. These are called T-independent antigens, and the reason you care about them is because as healthcare practitioners, you may be asked to give vaccines to both young people and older people, and they may be different. And you want to ask yourself, why do I have to give the 1-year-old one vaccine and the 50-year-old a different vaccine? And the reason is because of T-independent antigens. So here's our B cell, and we've kind of exaggerated the receptors. And you see that this particular antigen is wrapping around the B cell and binding to multiple receptors, IgD, right? When this happens, when it's a big antigen with repeating epitopes like this, if it binds to a whole bunch of the receptors, it can actually, that's enough to get the B cell activated without a T helper cell. So that's why we call it T-independent B cell activation. Because there's no B cell involved, it's independent of a T cell. It only requires the B cell. And you care about this because there are quite a few actually of these kinds of antigens, particularly for bacterial capsules, so things like polysaccharides that have lots of repeating structures. And it turns out that this is all well and fine for you and me, but children under 2, their immune systems actually can't do this yet. And so if you give them a vaccine that has this kind of antigen in it, their immune system won't develop an immune response. So we actually have to do some fancy things with these epitopes and turn them into a different kind of vaccine to stimulate a T-dependent response. And it's less important that you understand the intricacies of that process, but more important that you understand that there's actually an immune system reason for why we have, in some cases, different vaccines for older and younger people. So under 2, this process doesn't happen efficiently, and we have to develop a different vaccine that stimulates the immune system differently.
It doesn't hurt him, it just doesn't work.
It just doesn't work, yeah. But you know, they're the ones who need the protection the most usually, so. Okay, and if you're interested in learning more about how you would develop this vaccine, I'm happy to go into the details, but if I tell you it won't be on the exam, most people will say, stop talking then. Okay. I want to stop for a moment and talk about immunological memory. This is actually a really important thought that we should talk about right now. And it's one of the reasons why vaccines, this is a good time to mention why they're so important. When you first get exposed to an antigen, your innate immune system gets going right away. But your innate immune system is not nearly as effective as your adaptive immune system. The problem is your adaptive immune system is really slow, really slow. And you know this if you've been sick for 10 or 12 days with a cold. You wish it would get going quicker, right? So when you first get exposed to an antigen, here's 14 days before, remember I said IgM gets made first and then it's switches to IgG. So 14 days before the IgG in your system peaks. This is our antibody titer. Okay? This is how much antibody you have in your system. All right, so let's say you get vaccinated on this day. Hopefully you don't get polio, right, until day 14. So this is why you got to stay protected from these pathogens until your vaccines get a chance to get going. Then we decide that this isn't really enough of an immune response. We're going to give you a booster shot. You've heard people getting a booster shot, right? So we vaccinate you again at day, looks like about day 21. Actually, I'm not totally sure. Maybe it's day something else. But you see that after you get exposed again, boom, really fast, you've got tons of antibody being made. That's because you have all these memory cells from the first exposure that can get activated much more quickly. And that's the beauty of vaccines. So what you want to do is get enough boosters so that you have a nice high titer of antibody, right? And that way, if you get exposed again, you'll have enough in your system that you'll be able to fight it off really fast. There are some pathogens that kill really quickly, long before your adaptive immune system has time to kick in. If your adaptive immune system is primed, you're going to be in much better shape, much less likely to get sick or die of these infections.
So is a booster the same thing as how the new HPV one, you get 3 different times?
You get it twice now.
It's 3, I just got it a couple weeks ago.
That may be for adults.
Okay, right.
Yeah, because my daughter got it 3 times, and then they changed it to 2
Okay.
times for kids who are under 14.
But yeah, because I have to go
Yeah.
back in 3 months and then 6 months.
Yeah, so that's just because, I'm sorry to say, but you're not a teenager.
Okay.
Not a teenager. See how I tried to slip out of that? Okay, yeah, no, you're not a teenager. For teenagers, their immune system's actually, you know, that's the thing, is as you get older, you need more higher doses of vaccines to have more of an effect. Yeah, my daughter was really ticked because they changed the guidelines right after she had her 3rd shot. And she felt really ripped off, which, it's a painful shot, yes?
Yeah.
Yeah, so she was unhappy. Okay.
Yeah, it hurts way worse than flu.
It's way worse than flu shot, yeah. Okay, all right, so immunological memory. All right. So B cells protect you from pathogens inside your cells or outside of your cells? Which is it, inside or outside?
definitely testing of the.
You can, yeah, so if, like a pregnant woman, they'll test for measles, mumps, and rubella, because rubella causes horrific birth defects, and they test for chickenpox too. So they test for your titers, they look for antibodies in your blood, to see if you're still protected. All right, so B cells, actually, I need to see the question. I hope it was B. They protect, now I can't get this to move. Outside, yes, you, okay. Outside of your cells, that's correct, okay? So B cells protect, because the antibodies can't just get inside your cells. They have to protect you from things that are already outside of your cells, okay? All right, here's the study guide question. What's the difference between B cell dependent T cell dependent B cell activation and T cell independent B cell activation? Do you remember that? A T independent antigen looks like what?
The line with the.
It's long, with lots of repeats, and it can wrap itself around the B cell to activate it. Okay? T cell dependent B cell activation means that you need to have a T helper cell in order for the B cell to be activated. So the answer is in the name. Okay? So if it's dependent on a B cell, it requires the presence of a B cell. If it's independent of the B cell, it's one of these long molecules that is repeating epitopes. It can wrap itself around the B cell, bind to multiple receptors, and activate it without a T helper cell. Okay. So the last part of what we want to talk about is the difference between a CD4 and a CD8 cell. CD4 cells are T helper cells, and I'm going to use these terms a little bit interchangeably right now because that's what you're going to hear in the clinic. Over the years as the HIV crisis sort of raged on, one of the ways people differentiated between cytotoxic T cells and T helper cells was refer to proteins on their surface. So a CD4 positive cell is another way of saying a T helper cell. A CD8 positive cell is a cytotoxic T lymphocyte. And this is not a helper cell, this is a killer cell. It's an assassin cell, it's a murderer. Okay, try to keep you awake. It's violent. All right, so CD4 positive cells, which is what we've been talking about too, get activated by binding to MHC antigens on an MHC class II receptor. So here's our antigen presenting cell. It's presenting an antigen to the CD4. CD8 positive cells are cytotoxic T cells. They actually bind to damaged or infected cells. So this is not a professional antigen presenting cell. This is just a poor, normal cell in your body. It's had the misfortune to either become a tumor cell or become infected with something. And so what it's doing is it actually takes what it's infected with and kind of as a last gasp, it chops it up and it presents it on MHC class I. Remember that all of your cells have MHC class I? So MHC class I is like all of your cells are able to put something on their surface and say, hey, I'm so sick, come help me, come find me. They're actually not saying come help me, they're saying come destroy me. Come get rid of me because I have bad infectious virus inside me. So these are really different, right? This is an antigen presenting cell, a professional. This is a sick or tumor cell that's not a professional antigen presenter, but it's doing it in a final last act of desperation to get destroyed before it kills the whole organism. And so if CD8 positive cells bind to MHC class I receptors that are presenting antigen, and it's a way for them to know that a cell's been damaged so that it can destroy that cell. All right. I don't like reading slides full of text, but I do like you to have these for their notes. All right, so let's go to, all of that stuff is important, but we're going to cover it as we talk. All right, so what happens with a virus-infected cell? So this cell is got MHC class I. This is a liver cell. It's infected with hepatitis virus. How about we say that, since we've been talking about hepatitis? Here's our liver cell. It's got hepatitis virus, and it's going to process the virus, and it's going to present it on MHC class I. It's got to tell the immune system somehow that it's infected and needs destruction. All right? And what happens is that this virus-infected cell is going to bind to a cytotoxic T lymphocyte or a killer T cell. And this killer T cell has already been activated by a T helper cell. So this didn't happen by itself. It requires the chemicals that are present from the T helper cells in order for these to become active. So we don't see this because there's no direct binding, but it's been activated by the T helper cells that are present in the cell, in the organism. So this cytotoxic T cell binds to MHC class 1 with this antigen. It says, oh dear, you are infected. Say goodbye, right? You think of one of those like Arnold Schwarzenegger movies. And then it's going to do the same thing that a natural killer cell does. It's going to take perforin and poke holes in this infected cell and then secrete these granzymes that are going to go in and cause the cell to self-destruct. So it's taking out tumor cells, it's taking out infected cells. These are very important cell types in your body in terms of destroying infected cells. Okay, so this is basically just asking you to explain what I just did, which is that killer T cell kill cells basically the same way as a natural killer cell. It's going to secrete perforin to poke holes inside the cell, and then it's going to result in granzymes going in that are also secreted and causing it to undergo programmed cell death. So I have a couple videos here. This is just showing T cells killing cancer cells. So this is the idea of programmed cell death. You know, it's a nice shape and suddenly it balls up into little bubbles and dies. This is cell killing happening here. It's a cancer cell, so it's okay. So healthy cell, oh, been destroyed by a T cell. You could watch this over and over, right? I have another good one here. So killer T cell, the cancer assassin. So the way these kill cancer cells is the same way they kill a cold cell that was infected with a cold virus. It works really the same way.
Inside all of us lurks a serial killer. A killer whose primary function is to kill and then kill again. These are cytotoxic T cells, a specialized member of our white blood cells. They control our bodies, identifying and destroying virally infected and cancer cells, and they do so with remarkable precision and efficiency. There are about 5 million T cells in a teaspoon of our blood, engaged in a ferocious and unrelenting battle to keep us healthy. These amorphous globs move around quite rapidly, pushing out their leading edge and probing their environment as they go. When a cytotoxic T cell finds a cancer cell, membrane protrusions rapidly explore the surface of the cell, checking for the telltale signs
See how
of
it's
cancer.
crawling all over it, looking for it?
They kill their targets using poisonous proteins, visible here in red. These cytotoxic granules move down special pathways in the cell called microtubules to the interface between the T cell and the cancer cell. The T cell punctures the surface of the cancer cell and delivers its deadly cytotoxins.
The granzymes.
This is very important in our bodies, where cells are packed together, as it focuses the lethal hit on the target and minimizes collateral damage to the neighboring healthy cells. The fate of the cancer cell is sealed. The T cell then moves on, hungry to find another victim.
What do you think? Cool, right?
So is there already immunotherapy they can use then to like figure out what your cancer cell is and send you more?
Yes, this ends up being really complex. So we already know that T cells bind to specific targets. So a big strategy in treating cancers and cancer immunotherapies is so one thing you do is you can take T cells, killer T cells, from people who have the cancer already and figure that you have some of these cells in you, and if you isolate those and infuse them back at high levels, that you'll be able to put. So that has worked for some people, but it's really expensive, right? Because you can't just give someone a drug, you have to actually isolate their cells and clone them up and figure out which ones respond to their cancer cells. So these are really expensive. There are whole other sets of drugs that people are, immunotherapy type drugs that they're putting out there. So they think that some cancer cells put out these checkpoint inhibitors so that basically cancer cells are telling the T cells to shut down. Don't divide, stop, don't come after me. And so if you can find drugs or infuse people with T cells that aren't affected by the checkpoint inhibitors, or find a drug that blocks the checkpoint inhibitors, you can cause the T cells to be able to go back to do their job. So what causes cancer? Well, turns out it's a bazillion different diseases. It can be a mutation in how your immune system is working. It can be a mutation in a cell. Oftentimes it's a combination of mutations. Almost all cancer cells have multiple things that are wrong with them because your body has so many checks and balances to keep cancer cells from making you sick that in order for a cancer cell to actually be successful, your body's usually messed up in a bunch of different ways. And this is why cancer is typically a disease of aging. Because the older you get and the more times your cells have divided, the more changes and mutations and things have gotten messed up over time. Remember we talked about the spontaneous mutation rate in cells, and we said that every so often a mistake gets made and it gets carried over? Well, older people have way more mistakes that they've accumulated than younger people. Therefore, cancer is just way more common in older people than younger people. So someone who's 85 gets breast cancer, that's really a disease of aging. It doesn't mean they don't want to live and we shouldn't try to treat them and do what we can, but the point is that that's not uncommon. That's part of the aging process. It's kind of a natural outgrowth of how genetics works. We expect people to get cancer as they age.
So just to clarify, that the perforin is what pokes holes in the cell?
Perforin pokes holes in the
And
cell.
the granzyme is the pockets of poisons
It's the
that
pockets
gets
of
transferred.
poisons that go in and cause the cell to self-destruct. So I only have actually 3 more slides here, which is actually what I'm doing on time. By 1 minute. All right, so that's B cells and T cells in the adaptive immune system, okay? It's really important, so we're just talking about cancer. That's a situation where basically, in many cases, your immune system isn't actually killing the cancer cells for a bunch of reasons. It can be the cancer cells tricking the immune system, it can be the immune system not functioning right. The other half of the story is that your immune system is attacking you and hurting you. So the immune system, what you've got in your body are killers. And what you want them doing is killing the bad guys but leaving you alone. And you can get ill from either them not killing the bad guys or if they start to hurt you instead. See the difference? So when they're hurting you, when your immune system starts to direct its killer activity on your own cells, that's an autoimmune disease. And we know that actually you have some systems in your body to prevent autoimmunity. And one of them are these regulatory T cells, or Tregs. And again, I think you're going to get into the clinic and you're going to hear doctors say CD4, CD8, Tregs. Those may be termed because there are drugs associated with some of these. So I just want you to have heard it once. Regulatory T cells are 5 to 10% of the T cells, and we think that their role is to suppress T cells that might accidentally react with self. So that's an important job of a Treg. We also think that they might protect good intestinal bacteria, because those are definitely not self, but you definitely actually want them to be protected from your immune system because they're exhibiting microbial antagonism, and they're able to prevent the bad bacteria from colonizing you, right? And so we think that Tregs may also have a role in your microbiome, in that balance of your microbiome, protecting the healthy cells from the bad cells. We also think that